Hello, students. Welcome to this session. Today, we are going to cover the chapter number 9, that is Biotechnology Principle and Processes of Biology 12 NCERT. So, in today's lecture, we are going to mainly focus on the session 9.1, that is Principle of Biotechnology, and later two other sessions will be covered in the next classes. So, let's start. Disclaimer. So what are going to be the learning objective of today's class? So first we will understand that what is like difference between the traditional approaches in biotechnology and modern research. What changes have been taken place over the decade in biotechnology that today we have become so modernized that we can even make test tube babies. Second, we will understand the fundamental concept of genetic engineering and DNA manipulation, that how we could manipulate the DNA of an organism and how this genetic engineering has bring such a big change in humankind. Third and the last will be that we will see the applications of this biotechnology in medicine, agriculture, and industry. We are going to just touch upon these application because in more detail we are going to discuss in the next chapter, that is mainly about the application of biotechnology. So yeah, next this is the index. So this is the index that we're going to follow. First will be the introduction. Second will be the principle of biotechnology, that is 9.1 session of NCERT, in which we will talk about the core techniques used in biotechnology, that is genetic engineering and bioprocess engineering. Then we are going to summarize all the key points that we're going to talk about all in these slides. Fourth, we will see some NCERT based questions. Then we will see some keynotes that you're going to take with you. Okay. And last, we will see some interesting questions that you guys going to solve yourself. So let's start. So, what is the basis of biotechnology? As the name suggests, that it is the integration of biology and technology. In this, we uses live organisms or enzymes to produce useful products and processes that is in the favor of humankind. This biotechnology, using of this biotechnology approach, that using this live organisms or enzymes to produce this product and processes have been beneficial to human through various application. Haven't you seen your mother like pouring a tablespoon of curd into the pot of milk and leave it overnight, and when next morning we see it, it became curd? What it is? Yeah, it is biotechnology only. So some of the traditional biotechnology examples involve are, as I said, making curds, bread production, wine making, and this like usually involves microbe mediated process. As we see that in making curd, we use lactobacillus. In bread and wine making, production and making, it is basically Saccharomyces. Okay. This is a pictorial representation of approaches of traditional and modern biotechnology, that how time changes. This is in this we have shown that fermentation and all those things, curd making, and in this how time changes, and this biotechnology has become so impactful to human users that you know we can now develop a test tube baby in a laboratory. Let's go ahead. Now, what is modern biotechnology? So, in modern biotechnology uses genetically modified organism. What does genetically modify organism? So, the organisms in which there has been whose genetic material have been altered through genetic engineering technique are called genetically modified organisms. We operate this on a larger scale. It is not like a traditional method that we used for fermentation and all those things in small culture or something. In this, we operated on a very larger scale. It includes various advanced techniques that we are going to discuss in upcoming slides too. Here again is the pictorial representation of approaches of traditional and modern biotechnology. So now let's move ahead. What is the basis of, again, we are talking about biotechnology basis only. So what are the applications of modern biotechnology? First, as I have mentioned, twice or thrice that in vitro fertilization. Before understanding anything, you have to understand what is in vitro. So, in vitro is an experience or procedure that are conducted outside of a living organism in a laboratory setting. So, can you imagine now we can make test tube babies in a laboratory? Synthesis, DNA vaccine development. What is DNA vaccine development? They develop a sequence, a specific DNA sequence that target particular antigens, and that antigens, modulate, and that's not modulate, that triggers the immune system, which is the basis of vaccine development. Gene therapy and correction. Nowadays, we can alter the genetic of an human to cure a disease or treatment. Have you guys heard of CAR T? That is chimeric antigen receptor T cells. It is one of the most, it is used to treat cancer, and it is one of the most hot topic used in the science in today's world. As it has been mentioned in NCERT, that what is the that European Federation of Biotechnology has given a definition to, like, what, to this modern biotechnology, and this states in which they have combined this both the essence of traditional and modern molecular biotechnology. And this is that the integration of natural science and organism, cells, parts thereof, and molecular analogs for product and services. What does it mean? This is the integration, that is the combination of natural science. Here, natural science doesn't only focus on biology, it is other engineering fields too, that is chemistry, physics, and engineering things. Its combination with organisms, that could be full yeast, bacteria, some things, cells, and even parts thereof. Parts thereof could include DNA extract, RNA extract, and something, and molecular analogs, which is the engineered molecules that mimic biological functions for product and services. The product or services that is useful and that can be used by humankind. This is the pictorial representation which shows that what is the scope of biotechnology. In these figures, we can see this is something that we are going to talk in upcoming slides, but here we can see that there is a bioreactor shown here, then there is this antibody structure, then there is scissor, which is cutting the DNA that we're going to talk about, and many more. Now, what is the scope of this? So, the broader historical perspective of traditional biotechnology, it includes mostly like fermentation processes, such as, like, Asian method in which we have used to preserve food, making of beverage production like wine making and all those through natural fermentation. But this modern perspective has focuses more on genetic modifications and molecular techniques. This we see more, we have given more focus to the DNA manipulation and biotechnology tools which we can use to enhance organism capabilities. But the one thing which is common in both is that at last we need some bio-based products, biotechnology services like testing and research. Means it is basically we are gonna get some product and services which is useful to us. This is again the same picture that has been shown in the last figure, that what are the scope of biotechnology. Now, the main part came, that is principle of biotechnology. So, what are the core techniques of this modern biotechnology? So, if you will see, first is so before that you have to understand that this birth of modern biotechnology happens by the integration of two methods or two, engineering. One is genetic engineering, second is bioprocess engineering. First, we're going to talk about genetic engineering. So, genetic engineering is what? It is like deliberate modification of an organism genetic material that we forcefully modified or alter the DNA, that is genetic material of an organism, to bring a new phenotype in that organism, which involves we can either add, remove, or alter its genetic sequences. As shown in the figure, Like, see, in first figure, they have shown that they are working on some GMOs, that is genetically modified organism. Second is showing that viruses, how we could use viruses for genetic engineering, that we're gonna also talk about. That we are using genetic engineering mouse, cells, livers, heart. We can alter DNA using DNA, molecular scissors, and all those things. Cloning. Cloning is like making multiple identical copies. Clone, we used to heard like, okay, this is clone of this. This is the identical copy of this. So we can make the multiple identical copy of a gene. How, when, where, we will talk later. What are the key aspects of genetic engineering? First, as I said, we have to manipulate the DNA. Genetic engineering involves manipulating an organism's DNA, that the molecule that carries genetic information. We know this. Okay, that I will tell later. That we will tell later. So first we have to manipulate the DNA. Okay. Second, why we manipulate this DNA? Because we want in modification in some traits. Okay, in some physical characteristics, ability, or other traits. Third, recombinant DNA. So what is a recombinant DNA? It is a core techniques involve creating recombinant DNA which combines DNA from different sources. So recombinant DNA is a DNA in which the DNA, like, how to explain this? Like, okay, so it is a DNA in which there are two different part of DNAs, and those DNAs are together from, they are ligated together, but they are from the different sources. That you will understand more, like, in detail in upcoming slides. But here we have to understand that recombinant DNA is a DNA which have been made from the different sources. Here again, that diagram that we have shown in the last slide only, that is aspect of the genetic engineering, that how we can use this genetic engineering different things. Now, in this core techniques of modern biotechnology, what are the key techniques? So there are some key techniques in this biotechnology, modern biotechnology. First is the gene splicing. As you guys have already gone through that chapter, that is the molecular inheritance chapter. You know that the central dogma works like this: that DNA transcribe into mRNA, and then mRNA translate into protein, and then this protein is responsible for all the functions of the body. But when DNA transcribe into mRNA, mature mRNA, before that there is a stage of pre-RNA where there is coding and non-coding regions of this DNA. The coding regions are the exons and the non-coding regions are the introns. But we have to remove this introns to form a okay, so we have to remove this introns to form a mature mRNA. So while doing this process, this gene splicing can also be manipulated and used for this modern biotechnology. Second is the CRISPR-Cas9 gene editing. So CRISPR-Cas9 is one of the most hot topic in today's world. CRISPR stands for cluster regularly interspaced palindromic repeats. You guys don't need to understand this because it is not in NCERT and it is not even at the 11th, 12th level. But it is that bacteria's own natural defense system that make it possible that we can manipulate the DNA. Third is the recombinant DNA technology, in which we make this DNA, recombinant DNA, a DNA who are made from different source, who have two different source. Cloning, you guys know, that is making of the identical multiple copies. Then gene therapy. In gene therapy, the genetic alteration of patient happen to make it resistance or to prevent some treatment. As I have told, one of the gene therapy that has been came into trend nowadays is CAR-T, that is the chimeric antigen receptor T cell therapy. That also you guys don't need to remember or something, but yeah, it's just for the knowledge. What are the application of genetic engineering? So in agriculture field, there are several applications such as crop improvement and increased yield, disease and pest resistance, enhanced nutritional value, golden rice, this BT cotton, this BT brinjal that we're gonna study in more detail in next chapter, which is like biotechnology and its application. We are gonna, you know, focus more on it. So here we are just touching upon it. So yeah, this is the application of genetic engineering. A figure shows that how we can use in it gene surgery, drug development, animal models, genetic variations, materials, food, fuels, biofuels. You guys have heard of biomethane, bioethanol, and all those things. When we talk about medicine, so the applications are gene therapy that we like mentioned twice or thrice, disease treatment, drug production, genetic testing, personalized medicine. Nowadays, we have this new approach where we are saying that genetic profiling of people are different. So we are saying for different people, there should be different medicine. So using these things, we can, make a personalized medicine. Then industry, production of enzymes, biofuel development, biodegradable materials, chemical synthesis. These are the applications that can be used in the field of industry also. In research, study of gene function, that how genes work, what is the mechanism of the disease, what is the evolutionary biology of something, what is the development biology. We can also use this genetic engineering. Now comes the second part, the part which we have talked about, like without which the modern biotechnology could not be possible, is the bioprocess engineering. So, what it is actually about? So, it is for example, we have found that how to make a recombinant biology, but we have to apply some engineering principles to design, develop, and optimize the process for the production of that recombinant uh, DNA. And that's where this bioprocess engineering came into the picture. It helps us in making a microbial contamination free environment. It makes a sterile environment for us. It uses chemical engineering principles and different principles to design, develop, and optimize the process. It actually consists of two parts. First, upstream bioprocessing, and second is the downstream bioprocessing. In upstream bioprocessing, it is the initial stage of biomanufacturing. It is the initial stage of biomanufacturing cells or microorganisms that are cultivated to produce the desired product. Okay. But the second, that is the downstream bioprocessing, it actually involves in the recovery, purification of the desired products which we want to bring into market. Okay. So, if we will see the component of the bioprocess engineering that I have talked like now only, first is this upstream processing, which involves steps like cell line development. If we are working on some cells to, we are taking some DNA from a cell. So first we have to develop the cell line of that organism or some whatever we are using. Then we have to optimize the media. We have to see that that media consists of every nutrients, every essential things that are needed for the cells to grow should be, so therefore media should be optimized. Third is the bioreactor design. What is bioreactor? So a bioreactor is a device which help us to create a men- sorry, which help us to create a control environment for biological reactions to occur. It will give us a environment where there has been a perfect optimum pH temperature in which a cell and cell or organism can grow. So this is the function of the bioreactor, and its design should be precise because it is very important to make sure that the aspect ratio of this bioreactor is fine. Like what is the aspect ratio of the height to diameter of this bioreactor? Okay, so this is very important. Then fermentation control. We have to see like the what kind of environment, everything we have to control, like when we are using this in this upstream processing, we have to make sure that every parameter is optimized. Because without this, we can make a undesirable product also. Okay, there could be some contamination or something like that, so we have to make sure that all these process should be proper, appropriate. Then scale up considerations, we have to see that how much, we have to scale up this up. Like, we can't make, like, we can't use any aspect ratio. We have to be in a condition that okay, we can use this aspect ratio only. We can scale up this, taking into consideration that okay, even scaling off of our product, this could not, contain some contamination or something. We have to make sure that this is again the same process, same picture, which shows the approaches of bioprocess engineering. Second is the downstream processing. It basically involves these things like cell separations. I said that it happens that okay, we got that um, product, we got the protein which we wanted, but now we have to separate it from the cells. So cell separation, product recovery, we have to recover a product, we have to purify a product, we have to make, after purification of the product, we have to see what is the formulation of a product, and that should be in optimum, like it should be optimal. Then we have to see the quality control, which is one of the most important step before taking our product to the market. We have to see all these steps very carefully. Without this, it could not be possible to take our product into the market. Here's the same figure that has been shown. So now, what is the equipment included in this bioprocess engineering? What are the equipments we needed during these processes? First is bioreactor. I told that bioreactor is a device which help us to maintain a control environment for biological reactions to occur. It has some optimum pH and temperature, which should be taken care of, which should be taken into consideration. Everything should be optimum. Second is the centrifuge. So centrifuge is a device which uses centrifugal force to separate substance on the basis of their different densities. Okay. Then it is filtration systems which uses, to filter out our product. Then is these chromatography columns which also use for the separation. Then some sterilization units to make our product free of like any contamination, microbial contamination or anything. This is the bioprocess engineering application of bioprocess engineering, which says that we can use this bioprocess in vaccine production, recombinant protein production, food industry, biofuel, sewage treatment, enzyme production, antibiotic productions, and many more. So now, okay, so it again tells us about the some applications that we can application of this bioprocess. Not only bioprocess but a whole this modern biotechnology is that we can use in the product, in the pharmaceutical production. Like we can use it to make medicines and all those things. Then we can use it in food beverage industries. Then we can use it in biofuel productions, wastewater treatment. We know that in wastewater there are a lot of organic materials, so we can use it. Then enzyme manufacturing and many more. And these pictures that have been shown in the earlier slide also. So now we have seen that what is a traditional breed. Okay, it should not be breeding, it should be engineering. What is the traditional biotechnology applications? Sorry for this mistake. And what is advantage of this genetic engineering over the traditional one? So here are the steps. Here are the points. First of all, it is more precise than traditional hybridization. So what is traditional hybridization? It is nothing but just like how the complementary complementary base pair goes and bind to its complementary part. So in the traditional genetic engineering, what used to happen, not only the desired part which we needed, but the other part, the unwanted part, it also got into the product. So it is therefore in this it is benefiting here in this way, that it is more precise than traditional hybridization. Okay. Second, as I told, it allows the introduction of specific desired gene only. If we, that is, if we want to add a gene which is, for example, a gene which is helping into antibiotic resistance. Okay. And now we want to incorporate it into some host. So it makes sure that only those gene which is making that, which is responsible for that antibiotic resistance, go and bind to that host for a host DNA and replicate and all those things. Only specific desired genes go and bind, and this replication it happens, and the only desired trait we get. But in traditional hybridization, it is like more like undesired trait also comes. Okay, so in that way, it is more advantageous. Avoid transfer of the unwanted gene. That's the same thing that I have told. It helps, it makes sure that there is no unwanted genes that go and transfer to the host, and that host replicated and that product has some undesirable trait or something. Okay. So now we will more focus on like what is the mechanism, or how it has, like how we've found, like how, what this recombinant DNA got found and all those things that we are going to talk about. Yeah. So first thing, what should happen is that alien DNA must integrate into the host genome to replicate. It means, as I told you, for making the recombinant DNA, we have to take the DNA from two different sources. So the alien source, that is the foreign DNA. Okay, the foreign DNA that we have took, that must go and integrate into the host genome. Host, what is host? Host is that organism in which we are going to transfer this foreign DNA. So we have to make sure that that foreign DNA go and combine to the host genome and then replicate also. It's not like just like that it go and you know bind to that DNA thing, but it has to replicate so that it can go to different, it can make into proteins, or it can go to the upcoming progeny. So how we should it, like we can make sure that it go and integrate into the host genome only and replicate. So we have to, for that we requires the origin of replication sequence. So what is the origin of replication sequence? So it is a sequence which we needed to initiate the DNA replication. This is the sequence only by which it can find and then start replicated. So we have to make sure that our alien DNA should go and bind near to replica ori, that is the origin of replication. And thus it is important also because, as I told, it should go and integrate into the host genome. It should be near the origin of replication sequence because this will make it a part of chromosome for inheritance. Otherwise it is not possible. First recombinant DNA. So first recombinant DNA has been formed in 1972 by two scientists, that is Stanley Cohen and Herbert Boyer. So how it became possible? So what they did, so they linked the antibiotic resistance gene, okay, to a native plasmid of Salmonella typhimurium. So now you guys should know that what is a plasmid. So plasmid is a autonomously replicating circular extra chromosomal DNA. It means it replicates, but it has nothing to do with the actually chromosomal DNA. It is an extra chromosomal DNA. Okay. So what happens? They link antibiotic resistance gene, okay, the desired gene, with this native plasmid of Salmonella typhimurium. And how did they got this specific antibiotic resistance gene? They got it using restriction enzymes, okay, also known as molecular scissors. So these are the enzymes which help, became one of the most important discovery, and it helps to cut the DNA at a specific location. That is the reason that they could take out the specific genes which are involved in this antibiotic resistance. Okay. And what they did, now they have took this antibiotic resistance gene using this restriction enzyme, and then they ligate it, means join it to the plasmid, native plasmid of Salmonella typhimurium, using DNA ligase. What is DNA ligase? It is an enzyme which help us to join DNA pieces. After, so now this plasmid along with this antibiotic resistance gene is what we call recombinant DNA. Okay. So now what happened? So this recombinant DNA got ready. What was the three basic steps in genetic modifications? First, we have to identify DNA with desired gene. In previous case, it was that antibiotic resistance gene. Then we have to introduce that DNA into the host organism. Definitely, after making that recombinant gene, we have to introduce it into the host organism for replication and all. And then we have to make sure that this maintenance and transferring of DNA to its progeny. Otherwise, there is no benefit. Okay. So, what are the key components in the DNA transfer? Means after the making of this recombinant DNA, which includes, I'm repeating it again and again, that is antibiotic resistance gene and the plasmid of native plasmid of this Salmonella typhimurium, we need something to transfer it to that our host organism, the organism that is going to replicate it. So now again a plasmid, which is the, as I told, autonomously replicating circular extra chromosomal DNA, act as a vector. What is a vector? Vector is a here vector represent the biological molecules that helping us to transfer this DNA into what? Transfer into DNA into host. As say that we will again restriction enzyme, we will cut the DNA, we will use DNA ligase to join the DNA fragments, and here the host organism was E. coli, which provides the replication machinery. And here we can see that how in restriction enzymes, DNA ligase, plasmid, gene guns, this CRISPR-Cas electroporation, these are the things that we're gonna talk, but in upcoming slides, in upcoming classes. What was the process outcome? That first we create a recombinant DNA, we allow the cloning of the desired gene. Okay. That we have to make the multiple identities of this desired gene. Then we enables these multiple copies of modified DNA. And this is the same picture that we have shown, that is the component of gene transfer. So what is genetic engineering? Here's this summary. So in this class, we have just learned till here that we have learned that what is the difference between the modern biotechnology and traditional biotechnology, and what is the key basic concepts of genetic engineering and the bioprocess engineering. So here is the summary. It involves the manipulating DNA to modify organisms' genetic makeup. We uses techniques like gene cloning, DNA sequencing, and CRISPR. We creates the genetically modified organism for various applications. Applications we have just talked about is that we use it in medical treatments, crop improvements, and pharmaceutical productions. For bioprocess engineering, we see that it primarily focuses on designing and optimizing biological production processes, involves fermentation, cell culture, and downstream processing. It uses bioreactors to grow microorganisms or cells. So here's the first question of NCERT, which which asks, describe the importance of vectors in genetic engineering. What are the importance of vectors in engineering? So vectors are essential. Why? Because they carry foreign DNA into the host cells. They allow DNA replication in host organisms. They contain selective markers for identifications. You're gonna know more about selective markers in upcoming slides, in upcoming classes, but you should, like, for a brief introduction, I should tell you that selective markers are the basically genes which is introduced in plasmids to identify and select cells that have successfully incorporated that foreign DNA to see like whether this, foreign DNA has been incorporated or not. Have multiple cloning sites for DNA insertion, can be specifically chosen for different hosts, okay, and enable gene expression control. It also common vectors include plasmids. I told it is a small circular DNA molecule that can replicate independently, that is autonomously in bacteria. Bacteriophages. This is the virus that infect bacteria and can be used as vector to transfer DNA. Cosmid. Hybrid vectors combining features of plasmid and bacteriophages that and specifically used because it can allow larger DNA inserts. Artificial chromosome, that is engineered chromosome, which is capable of carrying very large DNA fragments, and it is often used for cloning entire genes. Next question. Explain the concept of transformation in genetic engineering. What is the concept of transformation in genetic engineering? So, transformation is the process of introducing foreign DNA into host cells. Like, okay, it can be achieved through various methods that we haven't, I think, discussed in we haven't discussed in this class, but we are gonna discuss in upcoming classes. But for like brief introduction, you should understand that we can, introduce this foreign DNA into the host cells using various methods. First is the chemical method, calcium chloride treatment, which say in what happened we it has a procedure and we create small pores in cell membrane by making that cell chemically competent and allowing the DNA entry. Second is the electroporation, in which we uses the electrical pulses to create some temporary holes in cell membrane so that it can uptake the DNA. Microinjection, it can directly inject DNA into cells using a fine glass needle under microscope. Gene gun, so it shoots DNA coated gold tungsten particles into cell using high pressure. Viral, so we use this modified viruses as vector to deliver DNA into target cells. What are the keynotes of this, of this whole lecture? That is, first genetic engineering was conducted in 1973. The bioprocesses engineering dates back to Asian fermentation practices. Okay, that it is not something that we got to know today. It's been going on from decades. CRISPR technology has revolutionized the genetic engineering since 2012. Modern biotechnology contributes to 35% of global chemical production, and the human insulin produced through genetic engineering was first approved in 1982. This point should not be here. Okay, it can be here, but we haven't talked about it in the slides. But we will gonna talk about it when we talk about its application. That how we have genetically engineered the human insulin, and it got approved in 1982. And bioprocess engineering can help reduce environmental impact through sustainable production methods. Here are some interesting questions, okay, so that you guys gonna answer yourself. That first question is, what are the three key elements included in the definition of biotechnology according to EFP? Then you guys have to explain the roles of vectors in genetic engineering and provide two commonly used vectors. Then it is like, what is the importance of aseptic techniques in bioprocess engineering? And the last question is that what are the main considerations in scaling up a bioprocess from laboratory to industrial production? That you guys need to go and find it yourself. So here are some DPP questions that we are gonna answer. So the question first says, which of the following is not a core technique? Which of the following is not a core technique that enabled modern biotechnology? That in we have to choose one option from this that is not responsible for modern biotechnology. So first option says genetic engineering. We know that it is one of the most important aspect. Second is the bioprocess engineering. Here we also know that it is one of the most important techniques used for modern biotechnology. Third, say hybridization procedures. Yeah, hybridization procedure is not a core technique, whereas use of restriction enzymes, that are the molecular scissors, are important for this modern biotechnology. So here the answer should be hybridization procedure. We will save it. And next, what is the role of origin of replication in genetic engineering? So the first option says to initiate DNA replications. Yeah, we know the answer is first that we need origin of replication to initiate our DNA replication. But see, like we will can see the other options also. To join DNA fragments, we know that we needed DNA ligase. To cut DNA at specific sites, we know that we need restriction enzymes. And to transfer DNA into host, we know we need vectors. The answer would be to initiate DNA replication. We will save it and move ahead. Which enzyme is referred to as molecular scissors in genetic engineering? This thing has came up again and again, and we know the answer that the molecular scissors are restriction enzymes. So we will go ahead with it. Now, the question 4 says, what is the primary function of plasmid vector in recombinant DNA technology? That what is the primary function of plasmid vector? So the first option is to act as a molecular scissor. No, we know that restriction enzyme act as a molecular scissor. It is not a function of plasmid vector. Second, to replicate independently in the host. We know that plasmid is something that is can replicate independently in the host, but I don't think it's not. I don't think. I'm sure it's not the answer. Third, to transfer alien DNA into the host organism. That would be the answer because vector is something that is going to transfer alien DNA into the host organism, and then that host organism going to replicate. And fourth, to sterilize the bioprocess environment. No, this could not be the answer. The answer is third. Save and next. Last question says, why is traditional hybridization considered less efficient than genetic engineering? Okay, so what are the causes? Like in like what are the causes that make that traditional hybridization less efficient than genetic engineering? And here are the options. It cannot create recombinant DNA. Yeah, that is okay, but this is not only the reason that is less efficient. Second, it often introduces undesirable genes. Yeah, this is the reason that it could create the recombinant DNA, but it often includes undesirable genes. Third option, we will also see that it requires bioprocess engineering. Yeah, everything it's all requires bioprocess engineering. It cannot use plasmid vectors. No, the answer is second. It often introduces undesirable genes, and that is the answer. We will save it, and now we will submit this. And see, as we see, all the 5 options are correct. So yeah, that's all for today class. We're going to meet in next class and talk about the second session of this chapter. So yeah, thank you.